Yes, you want more QC, of course. And yeah, it's not, obviously, let's be honest, it's not so sexy, but it is very important. So we have to deal with it. It's a problem. And then we can focus with anything that's fun. Yeah, my name is Philip. I'm a data scientist here at SSI. I'm in the Metabolomics research group. I was asked to talk a bit more QC and also a bit about how we go about doing QC with the neonatal dried blood spots that you saw in the biobank. All right, so you've done your metabolomics project, your experiment, you use your QC sample. Hopefully you have some interesting things to show. Then your next problem is, yeah, now you want to tell me, how did you do it? So that I can replicate what you just did. But not only that, of course, that's the cornerstone of science. But you have to convince me that your data is good, so that I can have confidence in the conclusions that you draw from the data. And then you're going to have this problem, like we all do. That is, once you know something, you forget what it's like to not know that thing. So that's the curse of knowledge. That's why it's so difficult to teach. That's why it's difficult to give instructions. It's difficult to write a good paper so that people understand what did you do right. So it's a cognitive bias. We all suffer from it. And let me give you an example. This week, you're going to create molecular networks. And if you want to create a network, you have to connect stuff, right? So to a molecular network, we have to connect molecules. And we connect them based on how similar their fragmentation spectra is, as we get from mass spectrometry. And there are many ways that you judge similarity in mass spec data. But there is one that's quite successful, that's used first in this paper, I think, and that you're going to use probably when you do the networks, is this modified cosine score. So I wanted to write my own code for doing this. And only to discover that, no, I'm not going to be able to do that, because apparently this is so obvious, one of the algorithms, that you can't tell me what algorithm you use, only that it is a well-known problem solved by algorithms. Well, okay, so this is the curse of knowledge at best. But then you dig into it and you realize that there's actually a bunch of those algorithms. And yeah, it depends the rank of your matrix. It depends if you want an exact or an approximate solution. And yeah, this is fine if you could direct me to maybe the supporting information, which is more or less infinite. Or you could give me the code. But no. So don't be this guy. Because, I mean, we all suffer from this, but shame on us for not doing better. Because either you tell me that, okay, you think it's obvious, or you don't know what you actually did. Anyway, so that's the reason. It's a curse of knowledge. And of course, now you have to convince others about the quality also about your data, not only what you did. And I just want to, maybe this is obvious for people, but I want you to appreciate what we're doing in untargeted metabolomics. And these are a bit of conflicting goals. So on the one hand, we want to measure as many molecules as we possibly can, covering a lot of physical chemical space. And we have no idea about the concentration. So from an analytical chemistry point of view, this is a opposing goal, because we also want to be precise. We want us to be able to reproduce, to have reproducible methods, and it would be nice to be accurate, but we can't have it all here in untargeted metabolomics, and it's fine not being accurate because we mostly just want to see a difference. So we don't need to know what's accurate, but we want to know that we have some kind of precision. Yes. And so that's that. So I want to appreciate. So that's why this is kind of a discovery method. It's like you generate hypotheses. You don't usually have a hypothesis-driven research. You just want to discover, and that's great. And then on top of that, we have all the other problems with most analytical experiments. And so that is bias. We heard a lot about quality now, but no one actually defined quality. So, you know, that can be done differently, but typically it's some kind of spread in your data. And we want to catch biological variation, and we want to keep all the variation that's introduced while trying to measure that variation to a minimum. So low quality is where you just introduce so much technical variation that you don't see the biological signal anymore, and that's a shame. And of course, we have the biology from the beginning. So in humans, it depends when do you sample at the time of day, because we have circadian rhythms. That's a bias, for instance. Other extrinsic factors, so what people have eaten and not eaten, that's something we see a lot in the samples. And then there's the whole sample processing, because a metabolomic experiment should be a snapshot of the biochemistry when you sample. So after you sample, you want any metabolism to stop. So it means that the sample has to be stable. You don't want any enzymes to continue their work, so you have to make sure that you stop them. And also, how you extract the sample also, of course, dictates what molecules you end up measuring. So if you really want to do untargeted metabolomics, yeah, then you have to account for polar molecules, you know, fatty molecules, and so on. Then we have the instrument, and hopefully you got a sense that, yeah, there's a lot that can go wrong. And it's a bit of a diabolical method, because every time you make a measurement, like you separate your sample on a column, and you count the masses, the instrument makes contact with the sample. So every time you use the instrument, you're actually changing it. So this is such a big difference from, for instance, NMR spectroscopy, so it's more my background, where you don't make contact with the sample. It's all magnetic fields and radio waves, essentially. So you have to be on top of this. And then we have the data processing. You will see tomorrow there is a lot of different heuristics and models that goes into trying to make sense of the raw signals. Some are very model dependent, so that will bias your result. But that's why you have a quality management system, so that you think about all of this, and you try to minimize the bias wherever you can. So maybe you adjust for some drifts, you correct for multiple testing, for instance, in your statistics. You have QC samples, you make sure that the instrument is running to nominal performance, as you would expect it to be. And then also when you design your experiment, that you have a clear idea about how you're going to sample and how you work with those samples. So just wanted a bit of an overview, right, about what we were facing. So when you read a metabolomics paper, you start here, and then let the authors convince you that they've accounted for all of this. And of course, you can't say what is the one best way to do this, because it depends on the system, the lab, the instruments. And I can't tell you what best practice is, because I've only been doing this for 8 months. But I can point you to guys that know what they're doing. And it's these guys. It's the Metabolomics Quality Assurance and Quality Control Consortium, MCAC. If you only remember one thing from my presentation, MCAC. QC, Clinical Metabolomics, MCAC. And the guys that started this, they've been in the field for decades. So they've seen the field grow, which is fantastic, right? But they've also seen how the field diverge. And things are not always consistent. People don't really understand each other anymore. How to judge each other's work. So they are trying to harmonize. And also, like, what is best practice? Yeah, it's a moving target, but try to pool together the experience in the field and provide recommendations so that new people can enter it in a better position. And you should really take a look at them, because they have a lot of influence. I think they want to influence scientific journals to set up requirements for how you report metabolomics work and QC. Yeah, so it is important. Yeah, they have a lot of publications, and one publication, the latest one actually from last year, is how do you best report your data about quality? And they have a couple of resources. So during a project, they have a big checklist of QC related stuff that you can, as you go, you work through it. And then the idea is also that this is something that you could add to supporting information when you publish your work. There's a lot of lingo here in QC. There are many different QC samples, so we try to create some kind of resource about what we mean. So if I say a blank sample, that doesn't mean a lot. It's completely context dependent. And so it is with a lot of other QC samples. And then also some suggestions for journals. Well, I will not cover everything in this publication. It's too much. And some are the usual suspects when it comes to reporting methods, but there are some things that we can highlight, and that is the QC samples. So you have to tell me what type of QC samples, how you prepared it, and also what's the purpose of the QC sample. What do you want to measure? Is it, you know, within your batch precision, between batches, between labs, and so on? Then, yeah, there are tons of different QC samples. So you can't assume that you have a name for a QC sample and that everyone will understand what you mean. Instrumentation, yeah, the usual, you know. But important to you, so how do you calibrate your instrument? How often do you do it? What's the nominal accuracy? What's your acceptance criteria? And how do you confirm that it is? And ideally, you also report that. And then it's this concept of system suitability test. You report all of that. How often do you have to clean the instrument? Because after a while, it gets dirty. Do you know when you have to do that? And so on with the chromatography system. All right, and then the samples. This is a big subject, but yeah, you have to convince people, like, how do you make sure that your experiment is a snapshot of the biochemistry that you sample? And you have to make sure that the sample doesn't degrade, that it's stable, so that's storage. Temperature is important here, and if you freeze and fall and freeze and fall, that's going to degrade your sample. You have to report that, so you get a sense of what are possible events in your project where you degrade the sample. Yeah, this is something about the analysis with your instrument. Of course, how you run the program. How long does your measurement take? This can be important. How do you equilibrate your system? When? And what's your acceptance criteria for doing that? And we will look more on that later. Run order, because we have this drift effect, you also have to randomize the order, and how do you do that? And how often do you introduce QC samples? And so on. And then everything with data processing. So this is a big topic, but don't basically say you use default settings, because default settings will change. And how you analyze the data from your QC samples. And so what you have to report is some measure of variance, what's a spread in your data. And there are many metrics for this, but typically that would be the relative standard deviation, and you would report a kind of precision metric for each metabolite. And it's nice with the PCA, it gives an overview, but it's not really considered sufficient to demonstrate that you have minimal drift in the system. All right, so these are the samples that we have, that we typically analyze, these dried hotspots. And this would be, oh my God, I hope you can see it. But anyway, so this is 96 well plates. This would typically be the layout for a batch in a project. And so we would run multiple plates, and a plate is run in sequence, and that would be a batch for us. So we have, of course, our biological samples, and they would be case control samples, and they would be block randomized. Don't have time to go into that. We have process blanks to capture anything that's coming from the sample preparation. We have pooled samples, so that would be you take a small volume of each biological sample, create one pool, and then you distribute that throughout the sequence. And this you can do to create, to get a metric of precision within the batch. But also, if you have a big pool, you can use it over batches, and you can have a method-driven normalization of drift. But we can't do that for practical reasons. So we have to have I don't know, I call it pseudo matrix match QC sample. We use our blood on paper. That we can use as a reference between batches. Right, and now we are trying to step up our QC game. So not only we want to streamline the analysis, but we also want to standardize our QC reporting. So we hacked together a little app. It's mostly Python, and it's this Plotly framework. We use OpenMS to analyze the spectra, and now we're playing with MatchMS that we will see more on Thursday to match against the spectral library. So I'm very nervous because doing live demos is scary. Okay, so I was planning to show a bit of a system suitability test, and that is for the chromatography system. Data here. So this is the plate that you saw with the same QC sample run over and over. And this is the pressure that you see. This is the so-called pressure trace. Now, why does it vary, the pressure? Because it's very important that we have constant flow. That is something we want. But we don't measure the flow. You measure, it's only the pump, the speed of the pump. So I say, okay, I want this output from the pump, but you don't measure the flow. You measure typically the pressure of the whole system. So that's going to report that you have constant flow. But why does the pressure change? It is because we have a gradient. We mix our mobile phase. So this is the gradient. You change the proportion of solvents, and that changes the viscosity. And you want constant flow, but the resistance is higher, so you have to have higher pressure. So that's why you get this type of behavior. And if I zoom in here, I don't know if you can appreciate, but there's a rainbow here. The rainbow is the order. And if I put the slice, you see that it seems to go up and then slow down. So this is the equilibration of the system, that it takes a time. If you have a tiny leak after a couple of injections, you will put packings into place. You also have binding to the column. You have some sites that will bind to everything. It takes a little bit of time. And you can also look other traces where you see where we have a drift. So these are the chromatographic peaks. So you will see also that, yeah, it's a drift. So that's why before a project, you have to equilibrate the system. And ideally, you should show this also. All right, then we have our little QC check. Running over time here, Philip, so I'm stealing some time for you. Yes. But if we analyze one plate at a time before we go on to the next, and I will tell you how we do this. This is not in production, so this software we don't use it now yet, but the plan is that we will do it. So I'm going to load data from an actual clinical project, one batch. And then you have to supply the program with some kind of metadata. So it has to know, and in our findings, we have metadata. So is this a QC sample or is it a biological sample? You have to tell the program what is what, and typically you have some sequence number, tells the order of injection to the instrument. So that's the metadata you have to supply. And then the QC is based on looking for a set of compounds that we expect to see in our QC samples and biological samples. So we have a little library of compounds. This is not the whole library, it's just a subset, but it's just to speed up the computation a bit. So we have some amino acids, some isomers, for instance, isoleucine. We can put some nice data. And you can also add compounds if you want them. So what we're looking for is, in the data, can you find masses that are consistent with a given molecular formula that has a specific retention time window? And then we look for that in the data. So it's a accurate mass, accurate retention time approach, mostly. And then, as on settings here, we can do matching to a spectral library. So let's load a little spectra here, and now I'm really nervous. Okay, it's doing something. Yeah, it's relatively quick. But so this will be 16 samples, QC samples from a batch. It's a bit heavy now because it's running Serum also. Anyway, it's coming. Right, so here we get the summary table, so we can look in our long-term reference. So we look at extracted ion metagram peaks, and we look at the area, and we don't want that area to vary too much, so we have some acceptance criteria for that, and typically that would be 20%. That's like the consensus in the field. And then in our blanks, we don't want to have anything. So we look in our extraction blanks. There's some contamination, but it's very little. And then we can look in our pools, the baby samples. And they have caffeine, but it's not that the babies drink caffeine, it's the mothers, and we see it. Oh no, that was not the food. So I think I have to stop here basically. But I want to, we can also look at MS2 data. So here are the fragmentation spectra. And we can look hopefully in a library. No, we can't. Yeah. Okay, we have a bug. That's a shame. Anyway, I think I have to conclude here, but we're taking QC very seriously. It's a moving target, and hopefully we will share this at some point. But yeah, I forgot to say, so we're processing both the open data format, but also the data format from our instrument, which is a Bruker quadruple time of flight. Yeah, any questions? Yeah, but no more QC, I promise now. But it is important. Yeah. Yeah, let's do that. Yeah. Yeah. Yep. Yeah, I know. Thank you, Philip, for sharing.